Hello to Doctor and everyone watching this video. In this video, my friends and I are going to explain the defects in DNA repair mechanisms that can lead to diseases like cancer. In this topic, we will learn about DNA repair mechanisms and the diseases that can arise from defects in this repair process. So, what is a DNA mutation? A DNA mutation is a change in the nucleotide sequence of an organism's genetic material. Essentially, it's a typo in the cell's instruction manual. Why is it important? DNA mutations are crucial because they can alter the function of genes. How can it lead to cancer? Cancer often starts when DNA gets damaged, causing genes that control cell growth to get changed or mutated. Key genes like those that tell cells to grow, stop growing, or fix damage can all be affected, leading to uncontrolled cell growth. These mutations can occur in two main ways: spontaneously due to natural errors in the cell, or induced by environmental factors. These factors can include things like radiation from the sun or X-rays and certain chemicals in our environment.
DNA is constantly exposed to damage from UV light, chemicals, and even normal cell activities. To prevent mutations and diseases like cancer, cells have built-in DNA repair system to fix different types of damage. This slide shows repair mechanisms for single-strand damage. Base excision repair removes and replaces small damaged bases. Nucleotide excision repair cuts out large, bulky damage like UV-induced lesions. Mismatch repair corrects error made during DNA copying. When both strands of DNA break, the cell uses non-homologous end joining to quickly reconnect the ends. Through it can cause small errors. Homologous recombination is more accurate and uses a sister chromatid as a template for repair. Photoreactivation is a light-dependent repair where an enzyme called photolysis reverses UV damage like thymine dimers. Direct repair also includes enzymes like methyltransferase that remove harmful chemical groups from DNA bases.
Moving on, I'll be talking about defects in DNA repair mechanisms. So, what happens when DNA repair fails? When our DNA repair system don't work properly, damaged DNA cannot be fixed. This causes instability in our genes and increases the risk of serious diseases like cancer or disorders related to aging. Let's look at some examples. In base excision repair, mutations in enzyme like DNA glycosylase mutation, APE1 defect, and DNA polymerase beta mutation can stop the repair of small DNA damage. In nucleotide excision repair, if proteins like XPC or XPB and XPD are mutated, the cell cannot remove bulky DNA damage, like damage from UV light. Mismatch repair problems such as mutations in MSH2 or MLH1 cause small DNA copying errors to be missed. More examples, homologous recombination defects like BRCA1 or BRCA2 mutations are linked to breast and ovarian cancer. Non-homologous end joining issues like missing carrier proteins can affect DNA break repair. And lastly, the direct repair may fail if enzymes like MGMT are silenced or missing.
There are many diseases that are caused from DNA repair defects. For example, base excision repair defects are linked to neurodegeneration, aging, and cancer. Problems in nucleotide excision repair cause disorders like xeroderma pigmentosum and Cockayne syndrome, which involve extreme UV sensitivity and premature aging. Mismatch repair defects lead to Lynch syndrome, increasing the risk of colon and endometrial cancers. Homologous recombination failure is tied to breast, ovarian, and prostate cancers. Non-homologous end joining defects result in SCID, chromosomal translocations, and cancer. Even direct repair mechanisms matter. Low MGMT levels can actually make cancer cells more responsive to chemotherapy.
As a conclusion, early identification and studies in defect of DNA repair mechanism is important in maintaining DNA integrity to prevent diseases. As the future brings better technology, the emerging possibilities of new gene therapy or targeted sequence will contribute to help treat occurring symptoms and help find a cure for DNA repair mechanism defects and their respective diseases caused.